All right, guys, so we've been investigating proteins and we've done a lot to kind of get our minds around what proteins are, but let's go into a little bit more depth now. So, as we've discussed before, proteins basically do the nitty gritty jobs of every living cell. We know that they're made up of long strings of individual building blocks known as amino acids. And as we've already identified that genes in our DNA, so remember DNA found in the nucleus, are what code for the proteins and their structure. So you have your genes, then you have transcription, translation. So amino acids contain an amino group, a carboxyl group, a carbon, and a unique R group. We've talked about these, but remember that this is kind of that core structure of the amino acid. All 20 amino acids have this same structure. It's this R group that makes it very special. And this R group is what decides how those amino acids are going to bind together in the primary structure to allow them to create different structures based on their organization. In your notes packet, you should see one of the first things is a box with a little bit of text, as well as a open spot for you to draw a diagram of generalized amino acid. You should be able to label the amine group, carboxyl group, the alpha carbon, and the R group. So I want to talk about the side chains now. Next to the chart that is below that on your notes, I left a big blank spot for you to be able to annotate and label about the side chains. And so these are all of the R groups for each of the amino acids. All of these amino acids are non-polar. That means that they are hydrophobic. Think about what that means. They can't interact with water. They have no charge at all. And so when we're looking at these, glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and proline, all of these are likely going to be amino acids that are going to be found on the interior of whatever protein is being made. Unless, of course, you're making some sort of like hydrophobic membrane or something like that that can't interact with water. But remember, water comes into contact with just about everything. So it's really unrealistic for us to be putting all of these hydrophobic amino acids on the outside. Okay. So annotate your notes to make it be known that nonpolar side chains are hydrophobic. They have no charge, and they're unable to interact with water. The polar side chains have a slight partial charge on the R group. What makes polar amino acids hydrophilic? So these are going to be because they have that slight charge. Oftentimes what causes that slight charge, not every time, but oftentimes it's going to be an oxygen atom. So when you're trying to identify if something has a polar charge or not, slight polar charge, you're going to look for an oxygen atom, and then you're going to look, is there a charge on that side chain oxygen atom? Make sure that you're not mistakenly looking at this, because that's part of that carboxyl group. That's not a part of the side chain. You're going to want to look at this side chain here. And if it doesn't have a charge next to it, then we're going to state that it's likely a polar amino acid. Because I'm not going to ask you to memorize all of these, but I do want you to be able to identify where they might be found. So next, we're going to look at these last 5. So these are electrically charged amino acids. So these are ionic because of their charge. These ionic R groups make the amino acid hydrophilic. Remember that charged particles can interact with other charged particles. Water is slightly charged, so it can interact with electrically charged molecules. Again, the way to identify this is looking at your R group and seeing, is there somewhere that it indicates it has an electrical charge? So this oxygen atom here, it shows that it has an electrical charge. As opposed to here, this has an oxygen atom, but it doesn't show that there's any charge to it. So these are electrically charged. These two are going to be negatively electrically charged, which actually causes them to be slightly acidic. And that's due to the extra hydrogen atom that's bound here. And then these are going to be basic charges, and that's because of this extra positive plus charge here. So what I'd like you to do for the next there we go. For each of the 10 following amino acids, use the flowchart. So in your notes, I've given a flowchart to determine the properties of that amino acid. Record your results on the table on the next page. So I've given you guys this table, and I'd like you to record those results. I'll do one with you, and then I'd like you to work through the remaining ones by yourself. So number 1, I'm going to use my flowchart. It says, is an amino acid? Do you see an amine group, a carboxyl group, and an alpha carbon? Well, let's see. Here's my amine group, here's my alpha carbon, and here's my carboxyl group. So yes, it is an amino acid. 2, find the R group. I've already circled this out for you. Do you see an oxygen atom? I'm going to look. I do not see an oxygen atom. No. So that tells me that this amino acid is a non-polar hydrocarbon, and it will be hydrophobic. So on my table here, I would write polar and hydrophobic. Okay. I'm just going to click through these remaining ones, and I'd like you guys to pause the video and work your way through identifying them on your own. I'll give you the answer key at the end. Okay, so go ahead and use my answer key to help you identify whether or not you got those right or wrong. And so the answers, nonpolar, polar, ionic, etc., you guys can look through those, but I'd like you to now indicate whether it's hydrophilic or hydrophobic. Maybe you've already done this, but if not, please take the time to pause and do so now. But what you should have been able to identify is that anything that's nonpolar is hydrophobic. Anything that's polar is hydrophilic, and anything that's ionic or electrically charged is hydrophilic. Okay? So remember, proteins are made of chains of amino acids. So let's just look here at this protein. If this is my chain of amino acids, so this is just say an integral membrane protein inside of a cell membrane, transmembrane protein, is this amino acid hydrophilic or hydrophobic? How do you know? So right here, this red one, is it hydrophilic or hydrophobic? How do you know? Well, since we know that water interacts with the outside of the cell membrane, right? This is a very aqueous environment. We have water here all the time. We're going to assume that it's probably hydrophilic. So that means that it's either electrically charged in some manner, or it is going to be a slightly polar molecule. Okay? All right, so in moving forward, we're going to look at how we go from a gene to a protein. So we've got these 20 amino acids, and so how do we go from coding from a gene all the way to a protein? And as you guys have already kind of looked at, I've given you this flow chart of transcription to translation and how these pieces kind of fit together. So we start with our gene here. Remember, DNA is going to be made up of A's, C's, T's, and G's, which then transcribe into mRNA. That mRNA is then going to be kind of edited. We're going to take out those introns, and we're going to release just the mature mRNA. That mature mRNA is going to bind onto a ribosome. The ribosome is going to read the mRNA in sequences of 3, the codons, and then each of those codons call for a different tRNA, and that tRNA brings with it an amino acid, a specific amino acid based on this sequence, and that's when the primary structure of a protein is formed. Okay? So in your notes, what I want you guys to understand is that genomics is going to be the DNA. That's going to give us our genotype, or I'm sorry, our genetic information, which we refer to as the genotype. But metabolic pathways are going to be referred to as our phenotype, some physical thing that is expressed. Okay? So we've already learned about this process. Amino acids are linked together by condensation to form polypeptides. So I've asked the question here, 500 amino acids connecting together would produce how many water molecules? I want you to pause here and just think to yourself, what was that condensation reaction model that we did? Remember, I gave you the pieces of paper, and we actually did this using glucose, and we went from a monosaccharide to a polysaccharide. I had you guys using water droplets. Think about what that might look like in an amino acid formation. So peptide bonds join amino acids. It's a condensation reaction, meaning that water is released when the bond is formed. So two amino acids form a dipeptide, and polypeptides are formed from more than two amino acids bonded together. So if these are our amino acids, right, so we've already got two bound together here. Here's another amino acid that we're wanting to bring in and bind it. So this is happening in the ribosome. We want to add these together. So we take the hydroxyl that's on the carboxyl group of this amino acid, and we take the extra hydrogen from the amine group and we remove them. And that's how that condensation, that removal of water, causes this bond to form, and we call this a peptide bond. Okay? So please take a moment, again, pause, and here's another drawing of how that happens. Go ahead and please draw this in the box of peptide formation in your notes. But do remember that there's a hierarchy of protein structure. So you have your primary structure. That's just the order of amino acids. That's protein sequence. You have your secondary structure, and you have your tertiary structure. They look like this. Primary structure is all of the amino acids bound together. Secondary is those amino acids kind of weaving and binding to each other. Tertiary is binding within that binding. And then quaternary is when you have multiple polypeptides bound together. So again, primary structure is the amino acid sequence. The amino acid sequence is coded for by DNA and is unique for each kind of protein. And the amino acid sequence determines how the polypeptide will fold into its 3D shape. So if we have even just a small, slight change in the amino acid sequence, this can cause a change to the protein and causes it to malfunction. For example, misformed hemoglobin, so this is a protein, causes sickle cell disease. So if we look at our DNA structure, if we look at our DNA here, or I'm sorry, our primary structure of our amino acid, at position 6, when we see a mutation in the DNA, we're going to see a valine instead of a glutamine. Okay? That causes this big of a difference in the phenotype of red blood cells. So I'd like you to go back and make sure that you can explain how a change in the primary structure can lead to a large impact on the protein's shape. Oh, I'm sorry, we're about to go here, actually. So explain how a change in the primary structure can lead to a large impact on the protein's shape. And so if we have a secondary structure, this results from hydrogen bonding between the oxygen of one atom to the hydrogen of another. That's going to cause this backbone of amino acids. It's not a backbone between the R groups, it's a backbone between the amino acids. The R groups have already bound. They've already formed that polypeptide primary structure. Okay? So we do this by forming what's called an alpha helix. This is a coiled secondary structure due to a hydrogen bond between every fourth amino acid. Okay? So it just kind of folds in on itself. Another type of secondary structure is the beta-pleated sheet, which is formed by hydrogen bonds between parallel parts of the protein. Okay, so anything that lines up and can form hydrogen bonds with itself is going to form a beta-pleated sheet. I'm going to stop there with secondary structures, and I will continue with a second video on tertiary and quaternary as well as functions of proteins.